Good afternoon, everyone. We're going to get started with our next session. This is the Changing Temperatures and Diet session. Our first speaker is Ella Parkinson, who's going to be talking about the impact of rising temperatures on development and brain size in larval American toads. Take it away, Ella.
Thank you. Okay, hi everyone. My name is Ella, and today I'm going to be presenting some of my PhD work, which looked at the impact of developmental temperature on growth and brain size in larval American toads. So just an overview of the presentation. I'm going to start with some background about developmental temperature in the context of climate change, and then I will go into the methodology of the temperature exposure experiment I ran in the lab. Then I'll show you my results about the effect of temperature on developmental rate, body size, and brain size, and then finish up with conclusions and future work. Okay, so as we all know, human activity is causing temperatures over land and water to rise in comparison to pre-industrial temperatures. And the extent of warming that we continue to see between the present day and the end of the century will depend on ongoing emission rates. You can see three different emission scenarios in this graph, but what we can see is that regardless of emission scenario, we are going to see temperatures continue to increase. And this means that American toad tadpoles will experience continually warming temperatures during their larval development, which is expected to have significant consequences on their physiology and fitness. So for this study, I was interested in the effects of increasing developmental temperature on three traits involved in fitness, which were developmental rate, body size, and relative brain size, which is brain size corrected for body mass. So exposed to higher developmental temperature, firstly, I expected to see, there we go, an accelerated developmental rate. This is due to the increase in metabolic rate that occurs when exposed to higher temperatures. Secondly, I expected to see a decrease in body size at the end of larval development. And this is due to the temperature size rule, which describes how in most ectotherms, when they're raised at a higher temperature, they end up at a smaller body size. And finally, I expected to see an increase in relative brain size. This is because a previous study in another lab found that when green frog tadpoles were raised at a higher temperature, they had increased relative brain size. So there are many possible fitness benefits and costs that these changes could have. For example, an accelerated developmental rate could be advantageous in areas affected by drought or extensive predation risk. However, there may be trade-offs associated with that smaller body size, as generally smaller body size is associated with lower survival, breeding, and overwintering success. Increased relative brain size could have positive effects on fitness, and this is because increased brain size is associated with higher cognitive capacity, which is the ability to acquire and process information in the nervous system. Moving on to my methodology. So I collected egg strings from a local pond in Guelph, Ontario in spring of this year, brought them into the lab where they were hatched at a common rearing temperature, and then separated into 4 different temperature treatments in replicates of 10 tadpoles. My coldest to my second warmest treatments were achieved using room temperature alone, but because we only had 3 rooms available and 4 treatments, I used heaters in a water bath for that warmest treatment. Okay, so for all 4 of my treatments, they cycled between a daytime and nighttime temperature. The present temperature treatment in pink was based on current average river temperatures in Guelph during larval development. And then the other 3 treatments were based on temperature data from the Intergovernmental Panel on Climate Change, or the IPCC. So historic temperature in blue represents pre-industrial temperatures. The projected warming treatment in yellow represents expected temperatures during larval development at the end of this century, based on the most conservative emission scenario. And that extreme warming treatment at the end in red was added to enable a more complete thermal performance curve of the relationship between brain size and developmental temperature in amphibians to be developed, as we currently don't have this in the literature. So these tadpoles were exposed to their respective temperature treatments for the entire length of larval development before being sampled at the late larval Gosner stage 42, which you can see here on the left, and it's characterized by the emergence of the forelimbs. So once they reached stage 42, 3 measurements were taken. Time from hatching to reaching stage 42 gave developmental rate. Body size was measured as snout vent length, which you can see here on the right. And then the brains were dissected from the head and weighed to give brain mass, and from that, relative brain size was obtained. Okay, so moving on to my results. The first thing I found was that those raised at higher temperatures reached stage 42 significantly faster than those raised at cooler temperatures. So as expected, higher temperature equaled faster development. You can see that in extreme and projected groups, they develop significantly faster than present and historic. I did just want to point out here that the difference between present and historic was not significant, but that's probably due to a very small sample size in that historic group. The reason for that is, unfortunately, we had an outbreak of a water mold disease during the experiment, and this resulted in significant mortality, and it was that historic treatment group that was the most severely affected by that, resulting in very few tadpoles reaching stage 42. The second result I found was that those raised at warmer temperatures were smaller at stage 42, which is again what I expected to see. So you can see here that extreme and projected groups were significantly shorter in terms of snout vent length compared to the present and historic treatment groups. So as expected, higher developmental temperature equals smaller body size. The last result I found was that was relative brain size. And I just wanted to explain a bit more about what that is and why it's important and how we actually obtain that value. So it's important to use relative rather than absolute brain size because it allows you to focus on the differences in brain size between individuals that cannot be explained by body size. So in order to obtain this value, we plot the relationship between brain mass and body size for all the tadpoles in the data set. Then we fit a best fit line, which represents the expected brain size for a given body size. And then it's the residual value between the line and each individual is what we use as relative brain size. So you can see some tadpoles had a larger than expected brain, which is a positive residual value. They're above the line. And then others had a smaller than expected brain with a negative residual being below the line. So this is how we can compare brain size between individuals that vary significantly in their body size. So as I mentioned earlier, a previous study by Emerson et al. 2023 found that green frog tadpoles raised at higher temperatures had increased relative brain size. So in this graph, you can see the temperatures increase on the towards the right of the y-axis, and you can see that those raised at 28 degrees had higher brain size than those at 14 and 22. So this is what I expected to see in the American toads. However, in my study, I found no effect of developmental temperature on relative brain size. So you can see here, no significant difference between any of those treatment groups. So moving on to what I concluded from these results. So firstly, I found that higher developmental temperature accelerates the rate of larval development, and also that American toads, at least at the larval stage, appear to follow the temperature size rule because they had reduced body size just prior to metamorphosis. Longer term studies would be needed to see what the fitness impacts of these are. For example, whether smaller larvae go on to have lower survival, breeding, or overwintering success as adults. My second conclusion was that, unlike what was previously seen in green frogs, high developmental temperature did not actually increase brain size in American toads. This could be due to species differences in the effect of developmental temperature on brain growth and neural investment. It could also be due to differences in experimental methodology. So this study in green frogs, they actually exposed their tadpoles to the same temperature for the first 4 weeks of development before then going on to switch into their treatments. And they also exposed them to constant conditions, so constant temperatures, whereas I used fluctuating regimes. And it's known that constant and fluctuating temperatures can affect physiology and growth in different ways. Okay, so I'm planning to repeat this experiment next year with some adjustments to build on our understanding of how developmental temperature affects brain development in these toads. So my first change is to collect the egg strings from a different pond. The pond that we collected from had a lot of fish in there, and it's expected that that's how the water mold disease was transferred into the eggs prior to collection. We're also going to trial some different treatments of the egg masses for that water mold disease to try and prevent mortality and increase sample size. Secondly, I'm planning to raise a subset of these tadpoles through metamorphosis and conduct cognitive testing on the juveniles. And this is to see if there is an effect of developmental temperature on cognitive performance. It's expected that animals with higher cognitive capacity will cope better with rapid environmental change brought about by human activity. So it's really important that we figure out if temperature has an impact on that and if that can be explained by differences in brain size. My third change I'm going to make next year is to use temperature data I collected this year from the toad breeding ponds themselves to design the temperature treatments, because last year I had to rely on river data. So this will hopefully result in more environmentally relevant temperature treatments. Yeah. And finally, I'm going to adjust the methodology a little bit to be more similar to the green frog study to see if I can confirm that there is a species difference in the two species. So I'm going to expose my tadpoles to the same temperature regime for the first 4 weeks of development before splitting them into their different treatments. And if I still don't see an effect on relative brain size after that change, I'm hoping to do an experiment where I compare fluctuating and constant temperature regimes to see if it is the fluctuations in temperature that are buffering that effect on brain size that we are seeing in the green frogs, or if there is in fact a species difference in how developmental temperature affects investment in the nervous system during development. And finally, I just want to thank my advisor, Fred Lethbridge, and my lab mates for their help, and also to staff at the university who helped me with the setup of the experiment, and finally to Greg Meredith, who gave me access to his pond to collect the eggs. And thank you very much for listening.
Thank you, Ella. That was great. Do we have any questions?
Yeah. Yes, hi, thank you. I was just curious to know, it seemed that you had talked about the weight or the size of the brain, and I was just thinking about the difference between growth and development. So are you also going to be looking at aspects of what developed in the brain as opposed to it's just pure size? Like, you
mean like the different brain regions?
Yes, yeah.
Yes, so we actually are currently working on looking at the contribution of different brain regions to the brain. So we have an undergrad working on that at the moment. And I'm also going to be measuring neuron number from these brains because it's been seen recently that total neuron number may predict cognitive capacity better than just whole brain size. So we're hoping to have those that data soon as well.
When you raise them from tadpoles, I'm presuming they came from the same kind of group of clusters, so likely were mostly siblings. When you raised them, were they raised individually in separate tanks, or were they raised as a group?
I raised them in tanks of 10 each.
Okay, and how did you feed them?
I fed them boiled lettuce.
Cool. And the only reason I asked that is I'm curious if there's any possibility that the competition and what they ate might have also influenced it.
Definitely, yes. We tried to, the feeding was just ad lib. So it was hoping that there wasn't too much competition for food. So if there was, if they were running out, we just always gave them more. But that's definitely an aspect. And also the effect of the disease was a big confounding factor. So I can't make any strong conclusions that it really was temperature and not the disease component affecting that as well. So, but yes, we tried to control for that by giving them as much food as possible.
Thanks.
Jordan is up at some point.
I think we'll stop there. Thank you very much, Ella. All right, our next presenter is Jeffrey Ethier, who will be talking about increasing seasonal temperatures reduces chorus frog calling activity.
Yes, so hi everyone, I'm Jeffrey. And some of this work is just because we experienced an unusually warm summer/spring in the Ottawa region. So it got me thinking, how did this affect the phenology of the species in the area? And I kind of give away the big surprising results with my title, but that's okay. So increasingly more, climate change has become something that we've been considering about with amphibian declines. I'm sure I don't have to tell you guys, but amphibians are one of the most endangered taxa in the world. Originally, here we go. From like the 80s to about the early 2000s, we were really concerned about like disease and habitat loss, with disease being that red bar and the green being habitat loss. And then along the bottom is the ICUN status. Now in the last 20 years, we've gone to see that there's more appreciation, at least, for the effects of climate change, and those mainly being increasing temperatures and reductions in hydro period, as well as kind of these stochastic big storms. And we know that seasonal and climatic effects affect phenology of these seasonal patterns of frogs, especially calling behavior, with usually warmer temperatures meaning earlier calling and just more calling in general. And this is particularly important to focus on because, especially in frogs and toads, calling behavior is directly associated with breeding. And that's how they defend their territories, attract mates, and basically know which species are in the area. So my big question was, how does temperature affect the calling behavior of the breeding frogs in the Ottawa area? So the important players, at least that I'm looking at, are the wood frog, the boreal chorus frog, the American toad, and the spring peeper. While they differ slightly in their life history traits and the way that they breed, with wood frogs being kind of explosive breeders that in our area do everything in April, to spring peepers who have a more elongated or prolonged breeding from March to June. These are all species that begin usually to call in March and April and do the vast majority of their calling during those 2 months. So my kind of hypothesis and prediction is that the plasticity, or the way that they change their calling activity, will be similar among these species. So how did I go about doing this? I put out a bunch of these audio moths, these tiny little green boxes that are about the size of a pack of cards, in 3 years during what I'm calling the early breeding period of these species. So from about the end or middle of March to the middle of May, at each of the locations. At each year, there was either 2, 3, or 4 sites that are monitored. And at each of these sites, I did 5-minute recordings at the top of each hour. And that resulted in a ton of data, over 10,500 files with more than 880 hours of data. If I was better at math and better with computer science, I might have done some automation, but I physically went through those files, opened them up, and saw which species were present in those audio or calling in those audio files. And then what I was looking at was basically temperature and calling behavior. So first I looked at the mean daily temperature by year, as well as the mean number of hours above 4 degrees, with this being a temperature point in which I particularly saw species beginning to call. And this is usually around the temperature that most frog monitoring programs use. Sometimes you'll see 5, sometimes you'll see 6 degrees Celsius, but around that time is when they start doing frog call monitoring. And then I averaged this by year between day of the year, or DOY, 60 to 100, which is basically the beginning of March to the middle of April. And this is what I'm calling the early calling initiation period, where once again, frogs will begin to call and usually reach the peak of their calling behavior. I did a day of first calling on each species, And just for the sake of time, I'm just going to show you the comparison between 2022 and 2024. Warning, I'm going to be saying years a lot in the next few slides. And then finally, I look at mean calling activity by year, average for kind of the entire period from March to May. And what I'm calling activity is just the number of hours in each day that a species was found to be calling. So 0.5 would mean 12 of the 24 hours there was calling. Okay, jumping right into the results. Mean temperature, I found that in 2024, there was significantly higher temperatures. Just to orient you along, well, here we go. Along the bottom x-axis is the year, and then the y-axis is mean temperature with a dotted line at 0 degrees, just to kind of orient you. And hopefully it's fairly apparent that there's a lot more temperatures, mean temperatures above that 0 line compared to 2022 and especially 2023. So was that significant? Yes, there was a significant difference among years, with particularly 2024 being significantly higher compared to 2022 and 2023. Okay, what about temperatures above 4 degrees? Once again, yes, higher in 2024 compared to the other years. Hopefully it's pretty obvious that, especially at the beginning of the year, from March to about the end of April, there was this big chunk of time or days where there was many hours above 4 degrees Celsius compared to relatively few in the previous 2 years. In 2022, they kind of caught up, but 2023 was pretty low. Was there a significant difference among years? Yes. However, this time it was 2022 and 2024 that were comparable, and then significantly different between 2024 and 2023. Okay, next. I'm particularly interested in chorus frogs. That's what my PhD research focuses on, so I'm kind of honing in on this species first. So I saw an 18-day advance in the calling behavior compared between 2022 and 2024 with, where is it? There we go. With calling starting in about April 10th versus the March 13th. So big difference there. Just on this graph here, we have day of the year of, there we go. It's not the greatest pointer. Day of the year, and then calling activity with a dotted line at 0.5. Once again, that's 12 of 24 hours in a day with calling. You might notice that there's 2 periods in which there is frozen ponds. So there is no animals calling while it's frozen versus the ponds were open, and then we do see some calling activity. The 2 things to kind of really note about this is that in 2022 and 2023, there's like a single chunk of time where we get a high level of calling with a steep decline at both ends of that versus this kind of roller coaster pattern, peaks and valleys in 2024, as well as an overall reduction in calling activity in 2024 with under 50 or 0.5 in 2024 versus the previous 2 years. And I found that was to be significantly different. Once again, 2024 is significantly different. Most interesting, sorry. And generally, if there's less calling, there's less breeding. It's not a perfect relationship, but that's usually if there's less males calling, there's less individuals and less breeding. So it's something to pay attention to. You don't have to look too closely at this graph. I just kind of wanted to show the various peaks and valleys and all the calling, and to show that in all 4 species we saw some form of advance in calling, with spring peepers calling about 9 days earlier, and then the highest amount being in my chorus frog of 18 days. But most interestingly is the relative calling activity was not different for the other 3 species, wood frog, American toad, and spring peeper, but it was for chorus frogs. So while there is plasticity in their behavior, it seems that only chorus frogs can't really seem to fully, adjust to this. There seems to be something else going on with chorus frogs that is not affecting the other three species. So just in general, yes, all the species are able to show some plasticity in the responses, but the response to increased seasonal temperature seems to be disproportionately affecting chorus frogs. Obviously, correlation isn't causation, and there could be a whole lot of other things going on. But considering these species are all occupying the same areas that are ephemeral, meaning that they're only wet with water for a small period of time and then very quickly dry up, and they're all in fierce competition, you expect that the relationship would be similar, but I'm seeing something a little bit different. There's other things that we have to look at to kind of really definitively say, oh, there's something being negatively impacting the chorus frogs. There's other aspects of phenology like the hatch rate, how quickly they develop and reach metamorphosis, as well as where they're migrating and how many are reproducing, which I have not been able to kind of present to you guys and are still unknown. But this shows that there is some form of impact on them. We have to investigate more what that is. And that kind of leads me into something I haven't presented today, but if you've been to other CHSs I've presented previously, which are some predictive models that include environmental factors other than temperature, as well as ecological interactions among these species. And with that, I would like to thank everyone here and to thank all my supervisors and all the people that have helped me throughout the years. And I open it up to questions.
Thank you, Jeffrey. So questions? Yep. Okay. It's on.
Yeah.
Okay. Yes.
Just wondering how you measure temperature, like what height, what were you using, just weather?
Oh, sorry. Yeah. The audio moths, the ARUs that I use, they measure temperature as well. So that's what I used. And I also compared it to the Environment Canada data just to see if there was ever like an error, and I got like almost perfect one-to-one relationship. Yes.
Hi. Yeah, great presentation. Would you just be able to repeat at what time they started calling in 2024 for the chorus frog?
The chorus frogs, they started calling at, I think it was March 13th.
March 13th? Okay.
Yeah, which
Give or
is
take. Yeah.
almost a month early.
Yeah. Okay. Thank you.
Awesome presentation. Thank you. Did you notice, and I know this isn't something you measured, but did you notice a big difference in the rate at which ephemeral ponds lost water among the 3 years?
Yes, unfortunately, that's something that I didn't measure, but just
like anecdotally.
Anecdotally, yes, they were much drier sooner. Usually they fully dry up around the end of June, and it was like the beginning of June.
Hi, I was just curious, you measured air temperature.
Yep.
I was wondering if you had any thoughts, speculations on the influence of water temperature in these ponds and when the breeding season may begin or have its peak.
That's a good question. I did have water temperature for it. Unfortunately, the units that I use, these little hobo units, once they reach a certain point, their battery kind of dies, and they have this like very weird, like it goes up and down. So I wasn't able to get like a full correlation between the two. But from what I could, there was also a very strong positive relationship. There's a little bit of a time buffer or delay, I should say, lag. So like when the temperature increased in the air, it took about 2 or 3 hours for it to kind of match within the water temperature.
Thank you.
Okay, we'll stop there.
Okay.
Thank you, Jeffrey. Okay, our next presenter is Rachel Fallas. Is that right? Rachel Fallas, who's presenting on ecohydrological controls on the resilience of snake overwintering habitat.
All right. Hello, my name is Rachel, and I'm here from the McMaster Ecohydrology Lab to talk about snake overwintering habitat. So our Canadian winters aren't necessarily very accommodating towards ectotherms like snakes, so they have to seek refuge from the cold by finding suitable overwintering habitat where they can brumate until conditions are favorable again. They usually establish hibernaculum below ground, taking advantage of rock crevices, tree root systems, or pre-existing burrows made by other animals. But in some landscapes, the availability of suitable overwintering habitat can look a little different. So I do my work in the eastern Georgian Bay landscape, which can be described as a mosaic of rock barrens, wetlands, and mixed forest. The landscape is generally lacking in deep soil deposits, and it's all underlain by impermeable granite bedrock. So that makes it a little challenging to find those deep crevices and burrows. But the region is considered a hotspot for reptile species at risk, and those reptiles turn to the peatlands when they're seeking a place to spend the winter. And they typically establish their hibernacula in raised mossy hummocks. Now, the research that my lab group has been carrying out was inspired by a mass mortality of eastern Massasauga rattlesnakes that occurred in French River Provincial Park. Essentially, the park employees set out one spring and found a bunch of dead snakes in their hibernacula after a winter that was especially cold and wet. And so that raised questions about how climate might influence snake overwintering habitat. The Georgian Bay supports some of the more stable Massasauga populations, but the entire species is facing decline throughout its range. In Ontario, these snakes can overwinter for up to 6 or 7 months of the year, and so they do need good quality habitat that can support them for that entire duration. And they also exhibit site fidelity in the winter, so they're not well equipped to cope with habitat loss and decline, which is rampant in southern Ontario. When we're talking about suitable overwintering conditions for these snakes, we apply this concept of the resilience zone. Some of you may be more familiar with the term life zone, but it's essentially this physical space within the hummock that's both unfrozen and unflooded. So basically it has to be between the 0 degrees Celsius isotherm and the water table. And over the course of the winter, that resilience zone size and position can fluctuate quite dramatically. With climate change, there are some concerns about how that's going to affect overwintering habitat. Some might assume that warmer temperatures would be a good thing because that would result in milder winters and reduce risk of freezing, but the reality seems to be a little bit more complicated than that. One of the consequences of warmer temperatures is going to be a reduction in snow cover during the winter time, and that means that precipitation is going to more readily contribute to the water table and increase risks of flooding. Meanwhile, reduced snow cover also means that the ground is less insulated against any sudden drops in temperature, and so we might counterintuitively see some colder hummock temperatures as a result of climate change. So the objectives of my research were to explore this threat and assess the influence of climate factors on the availability of overwintering habitat. And secondarily, I wanted to explore site-specific characteristics at different spatial scales to determine their influence on resilience zone dynamics. So as mentioned earlier, my research is based in Georgian Bay, and I collected data from 10 different peatlands distributed across these 4 different sites that are located along a 60-kilometer stretch of the Georgian Bay coastline. These peatlands varied quite broadly in terms of land cover and size in terms of what's available in the landscape, but Georgian Bay peatlands are generally quite small and shallow compared to peatlands from other parts of Ontario. At each of these study sites, we instrumented 5 hummocks and monitored them for 6 winters from 2018 to 2024. And we monitored the resilience zone by tracking the isotherm and water table. We calculated the 0 degrees Celsius isotherm depth by using thermocouple temperature profiles with measurements taken at multiple depths. And we also monitored the water table position by using a groundwater well. Each of these sites were also set up with a micrometeorological tower to monitor climate conditions such as precipitation, wind speed, radiation, air temperature, humidity, and snow depth. So we compared our 6 winters of monitoring, and we found that temperature metrics were most strongly associated with resilience zone size and availability. Each of the points in these 2 plots represents one of those overwintering periods, which we define as being from October through to May. And the error bars represent the standard error between the 10 different sites. So warmer temperatures were moderately associated with a larger mean resilience zone. So that suggests that warmer winters aren't linked to deeper freezing. However, temperature variability was significantly associated with reduced resilience zone size. So the outlook for overwintering habitat under climate change is a little bit muddled because we're expecting both warmer temperatures and greater temperature variability. Now, as a next step, we wanted to use the data we collected to train an ARIMA-X model that we use in conjunction with climate projections to try to forecast what water table and isotherm depth would look like under different climate change scenarios. Some of you might be familiar with the shared socioeconomic pathways, but they essentially outline different pathways under climate change. And these were the 3 that we looked at. So SSP1 is the sustainability pathway, whereas SSP2 is the middle of the road pathway based on our current trajectory, and SSP5 is the fossil fuel development pathway that involves intensified fossil fuel exploitation. So just a caveat here, our modeling was conducted at coarse temporal scales, and so it's not going to capture the full variability or the extreme situations, but what we're looking here for is sort of the general trends. And what we found is that there's a trend towards a shallower isotherm and shallower water table under SSP2 and SSP5. But I would like to point out that the magnitude of change for the isotherm over on the left does vary quite a bit depending on the site. And over on the right, there are actually 2 peatlands where we expected to see a deepening water table rather than a rising water table. When we put that together, we find that generally the rising isotherms will outpace the rising water tables. So that will lead to a trend towards increasing resilience zone size under SSP2 and SSP5. But for a lot of those sites, that change isn't necessarily very large. And there are still several sites where we're expecting a decrease in resilience zone size. And this is something that I am going to circle back to in just a bit after I talk about the ecohydrological controls on resilience zone dynamics. So we use field methods and GIS methods to obtain site-specific characteristics at the hummock, wetland, and watershed scales. And there were a lot of interesting relationships between these different variables, but the 2 relationships I want to focus on are these 2. So on the left, we found that the water table depth was significantly associated to the ratio between watershed area and wetland area. So a larger watershed area relative to the wetland means that there's going to be proportionately more precipitation being captured and funneled into that wetland. So that leads to greater water table fluctuations and increased risk of flooding. And then over on the right plot, we had a significant association between wetland size and temperature in these peatlands. And so we found that larger wetlands were associated with deeper freezing. Now, if we think back to the 2 peatlands earlier, where we saw a deepening water table rather than the rising water table like the other peatlands, we see that they're the 2 sites with the smallest watershed to wetland area ratio. And meanwhile, the sites that experienced the smallest change in isotherm depth were the sites that already have a shallow isotherm to begin with and are also some of these smaller peatlands. So when we consider all of this together, we see that peatland and watershed traits can indicate whether sites are resistant to freezing or flooding, and that is subsequently linked to the trajectories of those sites under future climate change. The sites that have the most positive outlook under climate change are the sites that are flood resistant and not freeze resistant. And then the sites where we actually see a decline in overwintering habitat quality are the sites that are not flood resistant, but are freeze resistant. If we contextualize this in terms of current suitable habitat, Massasaugas are likely to be using one of the shaded squares here. And so while the story here isn't entirely negative for those snakes, there is still some potentially suitable habitat where we might see declines in habitat quality. When we think about the potential implications of these findings, something that goes hand in hand with climate change is habitat change. Looking at these different wetland states, most Massasauga overwintering habitat is going to look like this towards the bottom, but if we're having water table rise, we might see these wetlands start to shift towards the higher wetland successional states. There are also all kinds of complicated feedback mechanisms at play in these wetlands. And so sustained changes over time could trigger runaway feedback processes that exacerbate those changes. Meanwhile, climate change is also causing an increase in the frequency of other kinds of disturbances like wildfire, which we got to hear about earlier today in the keynote and one of the sessions this morning. This here is actually a picture that was taken last year, less than a kilometer away of two of the peatlands that I was monitoring. And while the fire didn't kill this Massasauga, it did drastically affect the habitat, and that could influence the future survival of these snakes. And so here I'm going to plug a paper that a colleague of mine led, which was submitted just a month ago, that discusses some of the effects of wildfire on overwintering habitat for Massasaugas. Current habitat conservation priorities should include the preservation of peatlands that are resilient to flooding, because these are the sites that have the most positive outlook long term. But it's also a good idea to assess the distribution of overwintering Massasaugas across sites that are flood and freeze resistant to try and identify those populations that could be vulnerable in the future. And with that, I'd like to acknowledge the support I've received from my fellow lab mates and the collaborators and funders. I've been very privileged to be able to work on the traditional territories of the Anishinaabeg, and this work wouldn't have been possible without our collaboration with First Nations such as Magnetawan and Henvey Inlet First Nation. And lastly, thank you all for listening.
Great. We have time for questions.
Great presentation. I know that Chantel does a lot of work doing like habitat creation. Do you think that there's a possibility of like man making the right size hummocks in like with climate change coming in?
Yeah, so I've been able to look at some of the monitoring that's happened in actually peatlands that were constructed in developed areas. And something that there's been some research on is hummock transplants. Hummock size is actually something I didn't discuss in this presentation, but it's very important at the sort of smaller scale to help buffer against water table fluctuations. So that is definitely something with a lot of potential for creating habitat that can support these snakes. Yeah.
Have you considered, and I'm sure you have, but would you maybe help some of us, the value of flooding? Like obviously sites are made by wet environments and water stabilizes freezing processes and slows them down. So the anxiety about flooding, what if there's a real value to overwintering at least part of your winter submerged? It's not unheard of in the prairies for snakes to overwinter at the water table. So is the fear of the water table here maybe overblown?
Yeah, I think one of the things that is kind of missing from the research I've done is actually like how like the impact on the snakes themselves, because I am looking at pretty much just the habitat itself. And I know there's some research that has shown Massasaugas that have overwintered successfully, like being completely submerged for at least some of that overwintering duration. But I think, yeah, the real risk is that combination of flooding and freezing happening at the same time.
Really, it's really interesting. Like, this is really cool stuff. I'm glad you're doing it, but I just was wondering about the water, because water is important, and I've seen some constructed hibernacula that didn't consider that, and it very, very much worries me that we're going to get more of that. Thank you.
Thank you.
Excellent. Thank you very much, Rachel. Final presentation for this session number 8 is by Jordan Voss. Is that correct? Okay. And Jordan will be presenting on diet analysis of Alberta prairie amphibians.
Awesome. So my name is Jo, and I will be talking about the diet analysis of prairie amphibians, obviously. So I'll get into a bit of an introduction, some background in my study area, some research objectives, some of my methodology, the predicted results, hypotheses, and a couple of field preliminary results, as well as some significance, and then hopefully have some time for questions at the end. I will also say that all the photos that are in my presentation were all taken by me at one point or another. I absolutely love amphibians and have way too many photos. So as I mentioned, my name is Jo. I am a professional frog nerd, and I say that very proudly. I have a Bachelor of Ecosystem Management from Lethbridge College, and I'm currently working on my Master of Science at the University of Lethbridge. And I'm one of the very few people in the prairies that I know of that is actually actively studying amphibians. I am under Dan Johnson, who studies insects, primarily grasshoppers and crickets and that sort of thing. And I take any opportunity to take people out with me into the field, including my partner, Yella. So my passion started at a very young age. I would catch frogs and toads and salamanders on my family's farm and ranch. And I also worked as a park interpreter for 5 years, and through that position gained a lot of appreciation for sharing that passion with other people and getting them inspired about ecology and conservation projects, especially around wetlands and amphibians. So it led to my interest in my master's and my career because I was curious about the effects of pesticides on other amphibians, and because of that, I wanted to know more. And unfortunately, there isn't a lot of data available, and especially because I wanted to focus on the diet intake, there is very little information provided for the diet of Alberta prairie amphibians. So I led into this with very many questions and very few answers, and there are many knowledge gaps in amphibian data globally, and especially on the prairies and even for common species that you would not expect. And because, as we've all been made aware many times, they are one of the most at-risk groups of animals in the world. And I'm very interested in their overlaps of habitats throughout their life cycle because they do overlap between the aquatic to terrestrial phases and therefore likely come in contact with a lot of factors that could lead to their declines. So my study area is in southeastern Alberta and primarily on Alberta Conservation Association lands. So Chinook, escape coolie, and silver sage are the main 3 that I was working with during my field season this year. So I have Chinook, which is oh, no, hang on. Laser. There we go. This is Chinook, where it's a little bit more of an incised creek, and it has a very steady population of northern leopard frogs, which we unfortunately couldn't find this year. But you can kind of see me hiding in the bushes with all of my gear strapped on with my waders. This is by escape coolie with spadefoot toads, and this is one of the larger wetlands on silver sage. So my objectives were to compare habitat types of insects in their diet, so from aquatic subsurface to the aquatic surface and terrestrial. And originally I wanted to know more about the DNA of the actual prey items, but frogs really don't care about that. I also wanted to explore the relationship between amphibian morphology and prey size because I wanted to know at what point were they recognizing that this is a prey item. And I wanted to identify if sex and breeding timing affect the amount of food in their stomachs as well. So I used pitfall traps and drift fences in order to capture them. On the left is an image where they're all beautifully pristine set up, and the image on the right is when they put cows out. We did manage to catch a couple boreal chorus frogs in those specific traps, but we found it was a lot more successful to just go out with a beautiful butterfly net, very technical, very scientific, and to go capture them. And I had 2 field seasons where I unfortunately was not able to get data, so that led to a lot of anxiety where I'm like, I need to catch all the frogs now. So I do have a little bit of bias in that just because of how I captured a lot of my sample, but we did slow down and standardize as we went into the season once I had a little bit more data to play with. We used a couple of things in the factors to decide what we were doing for dietary analysis. There are three main standardized accepted practices where you have stomach dissection, which is where you kill the individual and then have to open their stomach to see what they're eating, which when you're trying to protect endangered species or at-risk species, you don't particularly want to be decreasing the population. So we needed something that was less lethal. There was the other option for fecal and DNA analysis, which also was not particularly conducive to what we were doing because it meant that it was a longer holding time for them to expel their fecal matter, as well as less cost effective in trying to do the DNA analysis. So we ended up settling on stomach flushing, which is efficient, quick, and non-lethal and relatively painless. I can get them down to a process time of about 1.5 minutes. So these images kind of show where we take the tube and we insert it into their mouth, and boreal chorus frogs are extremely tiny for those of you who haven't seen them. So trying to do stomach flushing on this kind of an individual is really intense. But we get it into their stomach and then we flush the contents out. I know this image is a little bit grainy, but we fed this particular individual 2 fruit flies in the animal care building so that we could see if it was effective. And we got all 2 fruit flies out, so it works. This is what the field setup kind of looks like where we're processing so that it goes through smoothly and we get it all figured out. You can see how excited I am to be out there. So my hypotheses are based around these 3 things. So dietary preference and range, that prairie amphibians exhibit a diverse range of dietary items, both terrestrial, aquatic subsurface, and aquatic surface species, reflecting adaptations to specific ecological niches and variations among the species. And I also hypothesize around the size and morphological adaptation that there's a correlation between the size and morphological features of the prairie amphibians and the size of the prey items that they consume that indicates an adaptive dimension to their feeding behavior. And finally, that breeding individuals and their stomach contents, that there is a difference in the amount of prey consumed by gravid and non-gravid females and mature breeding males. So field notes and preliminary findings. I was only able to capture boreal chorus frogs and male spadefoot toads. So it was really interesting, however, because gravid females of boreal chorus frogs did have smaller stomach contents, plugs is what I called them, more like less large plugs than non-gravid females. And generally boreal chorus frogs will aim for movement on the surface of the water while seeking mates. And we observed that while we were in the field. And this is an image from a video that I took while I was out there. And you can see that there are males calling right there, and you can see the ripples on the water and a couple of small bumps and bubbles. And those are all prey items that they would actively search and consume as they came closer. You can also see in the images on the left between the gravid and non-gravid females. We found a lot of pairs and amplexus, which was really cool for me because I haven't experienced that yet, and it was on my bucket list. And we also found a handful of boreal chorus frogs' eggs, which are really tiny, and we got to watch them develop for a couple of days. We also found that all of the plain spadefoot toads, the males, had empty stomachs when we caught them. And because their breeding is triggered by extreme rain events, they have one goal in mind when they first come up to the surface and find wetlands. And so their stomachs were all empty. And I find that really fascinating and hope to eventually get more data on that. We also found that most of the stomach plugs of the boreal chorus frogs have aquatic surface invertebrates, so things like flies, midges, and water beetles. And this is my most aesthetically pleasing photo of a spadefoot toad. He is gorgeous. So when we actually get into the lab with our stomach plugs, these are what they look like. They are covered in saliva, and this is due to the fact of how they actually forage. So when they take in their prey, they surround it with their saliva, which is a non-Newtonian liquid, which essentially means that it becomes more solid as they grip around it. And as they swallow it, they push with their eyes back into their stomach, for those of you who don't know how frogs eat. And it forms this plug that's in the stomach. So when we flush, generally you can tell that you have all of the contents by the fact of this is one thing and it's all come out. So then we start to pull it apart and try and identify what's inside of it. And when we look a little bit closer at it, we can see some more identifiable features, and they're generally pretty easily to identify as long as you know what you're looking for. I am not great at my invertebrate identification yet, but we did find some water bugs, a mosquito, some flies, water beetles, and spiders. And in some, we also found water mites, soil mites, and prey of very different sizes. So I'm really interested in what that will lead to for the actual statistical results. And lots of various bits that have somewhat digested or broken down. So sometimes you can find identifiable features and other times you can't, which is a little bit harder to work with, but we do what we can. So in significance, generally, diet is one of the main bricks for any life history information that you're working with a species on. And that can lead to things like their food chain protections and implications, especially when you start to think about things like pesticides and how they're applied throughout their habitats. I'm also eventually interested in how their nutritional value factors into their overwintering practices and how it leads to their survival. So I'm really fascinated and hope to work more into that topic in the future. Also, intrinsic value, I think they're neat. A lot of us think they're neat, but just getting other people to appreciate them as well and to realize that even though, for example, boreal chorus frogs are extremely common in Alberta, that doesn't mean that they're going to be around forever. So conservation and productivity of rangelands is a great way to get people inspired and into it, especially when you bring in the commercial factor that when you talk to a rancher and you say, if you have frogs on your land and you can keep your wetlands healthy, generally you're going to have better luck with your grazing in the future. And it gets them to care a little bit more. It also helps to recognize the factors that can lead to the presence or absence of amphibians within their habitats as well. So these are a whole bunch of my resources and not even an exhaustive list. I will now open it up for questions, and I'd just like to say thank you to a couple of people like Morel Alvarez, who's my field tech, Dan Johnson, my supervisor, Andrew Didiak for answering questions, and a whole host of other people who helped get me here and be successful. You can contact me as well if there's not enough time today with my email or LinkedIn, and that QR code leads to my LinkedIn.
Thank you, Joe. We have time for questions.
I'm just wondering, after you flush their stomachs, do you refeed them to be like, okay, I'll give you back your food? Or is it just like, don't let them go?
So they are voracious predators. So I don't intentionally feed them afterwards. I do put them back where I found them. So because we are processing in the field, that is the advantage that now that you're processed, I usually watch them for about a minute or two to make sure that they're responding appropriately. There's no shock. We did have a couple of instances with temperature where they didn't respond great. But generally when they respond well, you just put them back into their habitat. I usually watch them for a little bit just because I'm obsessive and they do go off and eat and do their thing and they're happy again. So I've not noticed any major implications for negative side effects. Thank you. Yeah.
So you said that none of the male spadefoots had fed at all?
Mhm.
That's neat, because usually they actually come up a little bit before.
Yeah.
Before they mate. They'll come up before the rain, eat, and then they'll go and breed. So
Interesting.
that's just really neat that they didn't have anything at all.
There was absolutely nothing in their stomachs. And it was interesting too, because it was literally the day after it rained. So this wetland was not there. It was actually on a road allowance. And we had heard them calling in the distance to go find them. And when we had found them, this wetland literally appeared overnight and they were there. So I don't think that they had enough time to really actively forage. They were just set.
Very cool. Thank you.
Yeah.
Does the time of night matter when you do the collecting? Because perhaps they call first and then they go forage afterwards. So do you have some kind of schedule when you go and sample?
So in my frog time, as I call it, we were out from dusk till about 4 a.m. So we were actively looking for them and checking for them to see if they were foraging. In this specific wetland, there was like 7 males and we could tell who was who. They each had individual markings that made them really easy to identify. And they were calling the entire night. Like I didn't see any difference to when they actually started foraging. So I'm not entirely sure when they actually switch, if it's kind of as they go or if they take a moment in between things or anything like that. I unfortunately don't have enough data, but really good question. I hope to answer that in the future. Do we have time?
One more question.
Can I speak to that for just a second too?
Yes, please.
Yeah, the breeding season for bombifrons is only like 2 days.
Yeah.
So they just go hard.
They're super cool. Awesome.
All right. Thank you, Joe.
Thank you very much.
All right.